I'm going to talk about the BioMAX beamline and how we can cater to drug discovery experiments. BioMAX is one of the beamlines at MAX IV, which Claire already described very well during her talk. So I'm not going to say anything about that. I'm just going to jump straight to BioMAX and what we can do. It is one of the two macromolecular crystallography beamlines currently in operation or nearly in operation at MAX IV. It's also capable of work with small molecules. The beam is very versatile. You can have a wide range of beam sizes and energies. From the point of view of industrial users, the nice thing about this beamline is the very standardized, highly automated experimental setup. This is a picture of the experimental hatch at BioMAX, highlighting some of the equipment we have. We have a humidifier and cryojet for different sample environments. The MD3 microdiffractometer is equipped with a mini kappa goniometer by default, but it can support other sample holders. The sample changer comes with a very high capacity UR with room for 29 packs. And the sample exchanging is rather fast, around 20 plus seconds. The detector is a first generation Eiger 16M. We typically operate it at 90 Hertz, so data collection is rather fast. And in a couple of months, hopefully, we'll replace it with a second generation version of the same detector, which will hopefully be able to go a little bit faster and also be more sensitive over the entire energy range of the beamline. For the software we use for experiment control and for sample management is we use MX3 and iSpyB, the iSpyB database, which are again standard programs in use at other facilities. All users at BioMAX have access to sample preparation laboratories, including a biolab. There are facilities for data processing in the MAX IV high performance cluster. We have Presto to provide a nice computing environment to run structural biology related software and macromolecular crystallography processing programs in particular. We also have facilities for data transfer, and we archive all data except proprietary data. So, but we give you the means to get the data out to your home institution. This is the website for more information about the beamline. The most common experiment by far is the standard rotation method data collection at cryotemperature. This experiment can be done totally remotely. The throughput is rather high. Okay, 3.5 minutes. You really have to go really fast and click buttons very quickly. 4 or 5 minutes per sample is more typical, depending on the type of data collection you want to do. Over the last year, we implemented fully automated data collection from well-characterized samples that don't need additional characterization to figure out the data collection strategy. We also support room temperature studies with a variety of sample environments. We can do in situ data collection, as shown in the image. As I mentioned before, we have a humidifier for humidity control, and you can also bring the crystals in good old-fashioned capillaries. For those of you who are still doing experimental phasing, we are fully capable of doing optimized anomalous scattering experiments, which are also useful for identification of metals, heavy atoms within the protein. And of course, we are also capable of more cutting-edge techniques like serial crystallography. And at this point, I should also mention the other beamline for macromolecular crystallography, MicroMaX, which is newly finished. So far, we've only collected data for one user group, so it's still very new. But this beamline will have enhanced capabilities for particularly for microfocus applications and for serial crystallography applications. It will be capable of doing time result experiments to the microsecond scale as opposed to the millisecond scale like at BioMAX. And if you're interested in this kind of experiments or in this kind of applications, I would refer you to Thomas Husby. So BioMAX is very well suited for drug discovery experiments. This is a big part of the scientific case for the beamline. So with even the standard setup is suitable for identifying and characterizing target molecules, even for very challenging cases like membrane proteins. It's also very useful, of course, for studying interaction with ligands, both high affinity ligands and also it's also because of the capabilities for automation and high throughput. It's very convenient for doing fragment-based drug discovery. Using lots and lots of small ligands with a non-affinity. The advantage of doing that is that, of course, you can explore a wider chemical space, and you can also find binding sites away from the canonical active site of the protein. So this is a very sought after application from users nowadays for both academic and industrial. So we've actually developed a dedicated platform for totally dedicated to this type of experiments, which is called FragMaX. FragMaX covers all the steps from crystal preparation to ligand analysis. The sample preparation is currently still done in collaboration with the LP3, the Lund Protein Production Platform, but very soon, as we speak, is moving to MAX IV, mainly for convenience reasons. The data collection, there's nothing special. It just makes use of the standard facilities at BioMAX. But these experiments are, I should mention that they are very well suited for the fully autonomous and attended data collection. So these days, FragMaX projects get often done like that with nobody actually sitting at the computer typing buttons. We also have a software FragMaX application, which is used for data analysis and optimized for identification of ligands. I don't know if Tobias is actually in the audience. I thought he had actually registered, but I didn't see him earlier on. But if you have any questions about FragMaX, he's the person to contact. So in summary, what you can get at BioMAX if you go the standard route, the standard experiments, you typically go from a crystal, a protein crystal, to a reduced set of structure factors. In some cases, you can actually get the electron density map. We have a pipeline that runs automatically when an anomalous signal is detected in the data, and we are currently developing a molecular replacement pipeline based on AlphaFold models. And hopefully we'll have that ready sometime this year. Then for FACMAX experiments, as mentioned before, you can actually jump in at the sample preparation stage. If you've already done your own sample preparation, then you can just use it for data collection and ligand binding analysis. In all cases, whatever the experiment, users are in charge of preparing and conducting the experiment. However, staff are available for support during the experiment, but also at the preparation stage and after the experiment during the data analysis stage. So you can contact us anytime and we'll try to help you. Regarding timelines and price, industrial proposals are not subjected to the same call cycle as academic proposals. You can submit them at any time, typically, but getting in touch with the Industrial Relations Office. The scheduling is rather flexible too. Usually, most beam times get allocated around several months in advance, but you can request beam time at any time, and usually, if you need beam time short notice, we can often accommodate that within 2 weeks, depending on what else is going on at the beamline. The price is currently that amount, 7,500 kronas per hour. And we allocate the beam time in shifts of 4 hours. The current industrial users typically use either 4 or 8 hours, but you can also get longer if you need it. And for more information, I would refer you to the Industrial Relations Office, who are also in the audience. This is the website. And I think I've used up my time. So thanks for your attention, and I can answer questions now or later in the breakout room.
Thank you so much, Anna.
I just want to show you a little bit how it works. If you don't have the expertise in your company to go to the beamline yourself, with the excellent help from the beamline scientists to collect data and then work with the data yourself at your company. But then you can access all the information through a mediator company, as we like to call us. And SARomics Biostructures is like that. So I just want to show you a little bit how we mediate access to large infrastructures. And in this case, it will be exclusively about the BioMAX beamline at MAX IV. So first, a little bit about our company, SARomics Biostructures. We started in Lund 18 years ago with the reason that we wanted to increase the use of structural biology information in drug discovery projects, because at the time in Sweden, this was very scarce compared to abroad. There were not really any ways to access the large-scale infrastructures in a easy way for industry, and therefore we started SARomics Biostructures. So we are more or less a contract research organization. We are still independent and still owned by the co-founders. we share and earn our money from zero activities, and then we invest that in staff and equipment, but also in some internal early stage drug discovery projects. We have a proprietary discovery platform, which in majority is about protein structure determination. But we also do quite a fair amount of fragment screening using a proprietary technology called weak affinity chromatography, but also lately crystallographic fragment screening using the BioMAX platform that was just presented by Anna. And of course, we also do NMR fragment screening, which you will hear from Carl later. We work mainly actually with foreign industry. Only a small part of our clients are from Scandinavia, majority from US, where they have a little bit different view on how to use structural information in their drug discovery project. It's quite a big difference in the view on why and how you use this information in an efficient way. So we work with mainly large-scale pharma and biotech companies solving small molecule complexes for small molecule drug discovery projects. But also, and this maybe is what actually sets us apart a little bit, we do quite a fair amount of antibody-antigen complexes as well for biotech companies, and then a small part of industrial enzymes. We are currently 28 people. Most of the people work with sample preparation, which takes almost all our time. So what we spend our time every day with is sample preparation. It's protein production and crystallization, depending on the different projects. And approximately half of the staff is working with protein production, and the other half is working with the X-ray crystallography, and a small part with NMR. And we have also sales offices in Boston, in Japan. And what do we offer? We offer wide range of all aspects of X-ray crystallography, everything from gene to structure. And I will also introduce to another service product which makes life easier for industry called we call them fast lane structures. And we also do a lot of antibody antigen structures, as I mentioned. And lately, NMR services is increasing a lot. It has been maybe a revival for that method in the industry right now, when peptides start to become quite a frequent modality for drug discovery projects. We also have add-on services where we help clients to use the data that we help them to generate. So we have a platform for computational chemistry where we use structure-based drug design. We have a license to the Schrödinger software. And I should also mention, we also have commercial licenses for all necessary X-ray crystallographic software that you use to determine structures. And maybe this is also a drawback for a small company that want to do it themselves. They don't have the necessary licenses, which is quite costly, I will tell you. We also use and help clients with full integrated projects that we usually collaborate with the medchem company in the same building as us here in Medicon Village in Lund. It's called Regularly Discovery, where they do the medchem and assays, and we do the structural biology and computational chemistry. And then the client comes to us with a target, and we deliver a candidate drug. And one example is, for example, the TAC1 inhibitor that Achillion licensed to Merck a year ago. That was Red Lead and SARomics that developed that one. And then we also do, as part of the integrated services, the fragment-based heat generation. Small part of industrial enzymes, and also, of course, our excess proteins are sold through a protein shop on our webpage. As I mentioned, we spend most of our time with sample preparation, and we have all the latest equipment for high throughput, low volume crystallization. So crystallization robotics, microliter robotics to dispense screens, and also plate to tell to image data automatically. And our go-to home source is, of course, the MAX IV laboratory, which is only located 2 kilometers away from our lab. And that gives us very flexible access because we can, to the last minute before we leave the door up at the MAX IV, we can add crystals. If we are to ship to other synchrotrons in Europe, we need to have a week or more in advance to ship the doors. This is very convenient, and it's our staff going there. We rent time, as Anna explained, proprietary time. And during a slot of 4 hours, we collect quite a number of data on quite a number of crystals. And if something goes wrong, we of course get excellent help from the staff scientists at MAX IV, which is very helpful. Lately, we also started to use the fragments platform, and there we need a lot of help. To simplify even more for industrial users and our clients, we have something called off-the-shelf protein structure. It's a concept where you have ready-made proteins, and the client just send the ligands, and in that way, you can in very fast way generate data with the ligands, because you have already the protein, you know exactly how to crystallize that and how to collect data. And over the years, we have gathered quite a pipeline or a platform of the different proteins, and this is common drug targets, and of course, quite a lot of focus on kinases for obvious reasons. But what we can say here also is that we have quite a portfolio now of common targets that are useful for antigen complexes, especially for biosimilars. Also want to mention, we have some here kinases in bold. They are proprietary targets. There are no structural data available in the public domain. And I just want to show you an example of one of these targets, and this is the IL-17 protein, which is a common drug target for antibodies, but lately it has been used quite a lot for small molecule drug discovery. And we have a proprietary crystallization system where we can generate small molecule protein complexes with this protein. The only previously published structure was taking help from actually a fab to chaperone the crystal formation, but we can generate this without that. And this was published last year by it's a collaboration with Leo Pharma, so it's our structures in this publication. And to give you a feeling for what this means, we actually have worked with 8 other clients with the same protein. So it helps a lot to have these common drug targets as off-the-shelf protein structures. And to give you also another example, but of an antibody-antigen system, we have another project with another Scandinavian company, Alligator Bioscience here in Lund. And we help them with to generate this data because when you develop antibodies, you need to define the epitope. Some companies rely on HDX data for this, but actually X-ray crystallographic data is a lot easier sometimes to generate, and it's much more accurate, and it costs approximately the same as well. So it's superior in defining the epitope down to atomic detail. Of course, you can also use the information for understand the mode of action of the antibody. You can use it early in the project for structure-based design, for affinity maturation, for example. And to some extent also it's used for higher order structure characterization for late stage products. And in this project with Alligator Bioscience, we determined two different antibodies actually that bind to different epitopes, and X-ray crystallography was used to define the epitopes. And in this case, it's an antibody towards the protein called 41BB, and antibodies towards this target help to activate T cells that specifically target the cancer cells. And in this case, we determined the structure of ATOR-1017 in yellow here, which appears to bind exactly as the natural ligand for this protein, which is visible here in beige color. And this was a single chain format of the antibody, but it helps to define the epitope, and also to compare it with other competitor antibodies that had a different mode of action, like utomilumab and urelumab. And it was shown that ATOR-1017 had a unique properties that gives much better safety and efficacy for this purpose. We also worked with another Fab that binds on a different epitope, and this antibody was actually useful for another purpose, where it is used in another format, in a B-specific format, with another Fab arm that targets 5T4, which is a common tumor antigen. So in this case, this B-specific molecule take the tumor cells and localize them towards the tumor antigen T cells, and then eradicate the tumor. And in this way, with this specific mode of action of this antibody, this is a much more effective process. And I think there my time is finished. So I want to finish with just some contact details. Thank you very much for listening.
So drug discovery and development possibility for industry at SciLifeLab NMR unit is part of the title for the webinar today. And when putting it together, I realized that it's not only the NMR unit that needs to be somehow mentioned and presented here. So I will also talk a little bit about the CBCS unit, because what we do is a service within that unit as well. So my name is Cecilia Persson. I'm the head of unit at the Swedish NMR Center, SNC for short, if I would say that later on. I work mainly with structural biology, which is part of our activities. So Wei Xiao and Ulrika, who I think are here in the audience, and also Matthias, who is a co-worker in Umeå, is the ones that are the experts and more hands-on people if you would contact us for this kind of project. So the Swedish NMR Center, as a SciLifeLab unit, we have different activities, which is structural biology, as I mentioned, where I mostly work. We do structure, we do dynamics and interactions. And the interaction part of the structural biology is what later on will be the example here for drug development and drug discovery. And metabolomics is a completely different area where you look at hundreds or thousands of samples, and you do instead a multivariate analysis, looking at statistics for the large cohort. Right now we're doing a long COVID study with between 6,000 and 7,000 samples, and hopefully we'll get some information out of that. We work with solid state pharmaceutical applications, and this is a usage of our latest investment, which is called a DMP NMR machine. And this is a very close collaboration with AstraZeneca, looking at tablet formulation and other questions for pharmaceutical applications. For example, also coming into living cells later this year. Small molecules is always on the carpet for NMR, but this is mostly local activities and not so much within SciLifeLab. So drug discovery, I will focus on talking about fragment-based screen, which is inherently built on one of the NMR characteristics that the larger the molecule, the size of the molecule affects the signal characteristics you obtain in your spectrum. So a small molecule will be behaving different than a big one. So when you have a drug or a fragment interaction with a protein, the fragment upon interaction will actually gain some of the characteristics of the interacting protein. And this will result in different spectra for your fragment once it is free in solution, here in red, or if it is bound to the protein. It can also lead to a change in chemical shift because the chemical environment for your fragment is different when it is bound or interacting with the protein. As part of the concept, you also can have a protein target, a control for your protein target, so that you have a competing small molecule or a fragment with the hits that you are trying to find. This is a very good thing to have for your target protein. It gives you a control experiment, and it gives you the possibility to investigate in more detail exactly how the interaction is done without getting real structural information about the exact site. And showing you briefly how the experiment is done, and what you need to actually go through with one of these projects. So all measurements are done in NMR tubes, of course, and for those of you who haven't seen it, they are small glass tubes, about 10 centimeters high and 3 millimeters in diameter, and we work with volumes in normal water phase at about 200 microliters. And you start out with your library of fragments. It can, and the total screen can contain a few hundred up to a few thousand samples, but to make it more efficient, we mix 10 and 10 in each sample so that we don't need to simply to reduce the number of samples we do. And the spectrum you record here, it's shown in blue in this example, is for the small molecule. And you will then, of course, have spectra from all your 10 molecules. Adding in the target protein will then, upon binding to one of the fragments, result in a reduced intensity for the compound that is actually interacting. So in this spectrum of overlaid spectra from 10 molecules, you can single out or identify those that are actually from the fragment interacting. And coming back to the inhibitor, or the already known binder to the protein, adding this one to a solution will actually then replace your molecule. And in the end, you go back to the initial spectrum as a control experiment to see that, in this case, that the fragment you identified actually binds to where the inhibitor binds. Of course, you can have binding which rearranges the protein, and then the inhibitor possibly doesn't bind this in the same pocket, but that's details which, I mean, we don't see them here, but that's details you can continue to study in other experiments. We record 2 or 3 different NMR spectra to have a good possibility to do the analysis later on. But as I will show you on the next slide, it's really important to also have this as part of a pipeline so that you don't just identify the hits and then you don't know what to do with them. So here it's again, I would like to mention this CBCS, which is the Chemical Biology Consortium in Sweden, a SciLifeLab unit. And I can't use the arrows. So here, to any drug development project or company, this is of course a known path on how you set up an assay design, and you screen and you find hits, and then you validate, and then you go and refine your hits. But what I want to say is that CBCS actually offers support for this pipeline of drug discovery. And for the screening part here, there are many different screens available, and NMR, which I'm talking about, is one of these. So it's a very close collaboration with this unit. Okay, so this is my busiest slide, and I wanted to reduce it a little bit with the details in how we come up with the price. So talking about time frame and timeline and what actually requires for a project. So if we start with what you actually need is roughly 2 milligrams of unlabeled protein if you have a 30 kD protein, and you need to have it about 0.1 millimolar or preferably higher concentration. So that's a prerequisite for actually being able to do the screen. The screen I'm talking about now is roughly 800 fragments. So you know ballpark how much it relates to. And you need 4 microliters of each fragment, and they need to be in a high concentration because they are diluted from DMSO to a water solution. And it needs to be in deuterated DMSO because otherwise we will see that the normal protonated DMSO will dominate the spectrum. So that's the prerequisites in terms of material. The final sample mixture is 5 to 10 micromolar of protein and then 50 to 100 micromolar fragments, so that you have a high access of fragment to, so even if you have a very low KD, you actually do get some binding of your fragment. And as I said, we usually mix 10 fragments per sample, but you can have between 8 and 12, of course. And then what's additionally involved in the price is spectrometer time, tubes, control experiments, and then, of course, some personnel time for sample preparation and data recording is also needed. And in the end, we end up with the price depending on how many experiments you need to run. For example, if you have fluorine in there, you can run additional experiments with fluorine. But we end up with a price of roughly 150 to 200 Swedish crowns per fragment. And then depending on the library size, it scales. And within a month, you can usually get time to measure your project with us. But we also realize that the protein production and preparing the things are more usually the time limiting step here. And planning the project, we do it together. Library preparation, that's mostly on our side to prepare and pipette and so on. And then the actual data recording is the smallest step, but it's still, that's one of the costs. And then in the end, of course, there's some data analysis needed as well. And then I want to mention the reversed screen. We call it screen, but it's more detailed study of the interaction. So once you've found your ligand, and this then requires that you also know the protein structure and you know the assignment for the structure of interest, you can turn this around, and instead of looking at the small fragment and adding the protein, you actually have your protein in there and add one fragment. And then what you can see is upon titration of your chosen fragment, you can see peaks moving. And in the end, you can also then, if you have the structure, map the interaction surface on your protein and get very detailed information. It says, so this is a titration with calmodulin, and anyone who worked with calmodulin knows that it has these 2 lobes. So this is not calmodulin, this is a different protein. And again here, this is a cheaper way of looking at the interaction since you only have one sample. And I will not go through the details, but the cost is between 1,000 and 4,000 if you want to run one protein with one fragment. And it's shorter in time. Summing up the things we can do within the fragment-based screens is ligand observed experiments and protein observed. And these 3 are my colleagues, Wei Xiao, Matthias, and Ulrika, who will help you out if you contact us. And then I wanted to finish with one example, our latest example of a screen that we took part. And it describes the whole thing going from a library to a single drug in the end, and targeting the main protease, COVID. And here it starts with roughly 3,000 fragments in the end, going through the whole chain, and then optimizing the ligand or the fragment and finding a good binder in the end. There's a reference here if you want to read about it. And then the very last slide is about developing, and what we are actually now starting or are developing at the moment is to look at your protein in the actual correct biological environment within the cell. So this would then require that you have an isotope labeled protein after purification and preparing your cells, whether it's mammalian cells or E. coli or yeast cells. It can be the system that you need. You electroporate in your purified protein, so you have it in the actual cell. And then as an addition to our NMR machines, we have equipment that keeps your cells healthy and viable by adding nutrition and so on for up to, I think it is 2 days. And that means that you can actually make some more long-term measurements on your protein within an environment where it naturally interacts with things there, or you can add your fragment even to this system. And then some contact information. Weichao, Ulrika, and me. I forgot to put Matthias here. I'm very sorry. And then our webpage is here, both the CBCS and Swedish NMR Center.
So my name is Khalil. I'm manager for NMR service at SARomics Biostructures, and I don't feel like I have the need so much to say anything more about SARomics, since Björn already talked about what we do. I should say in terms of NMR, we are basically doing what as a contract research organization, we are doing the same things as they do at the Swedish NMR Center for commercial clients. So we do fragment discovery, we do structural biology and dynamics. But that's not what I'm going to talk about. Now I'm actually going to move to something much larger, which is high-order structure assessment, comparability and characterization using NMR spectroscopy, which is something we've been doing for the last 7-8 years. And basically almost in all cases, we've been using the Swedish NMR Center for these spectrometers. So to start off, as you know, proteins are complex molecules with multiple levels of structure. Traditionally, if you want to look at the high order structure, you use indirect methods like mass spec, IR, CD, or DLS. They are kind of coarse measurements. In contrast, you have NMR, which is very sensitive to high order structure. Here's a rather extreme example where you have one available biodrug, filgrastim, in unfolded and folded state, and there's a massive difference in how the spectra looks like. And this is due since in NMR you report on the local environment of the atoms. So if we then take this, how can you then use this for studying antibodies? Well, as we say, if you look at an NMR spectra, it's a fingerprint of the 3D structure, which means that every peak that you see in the spectra is an atom or a methyl group or some form of, basically you have atomic resolution. But if you look at large molecules, sometimes you cannot isotope label them because they're not available to be expressed in a good strain for that. And then you have to live with the natural abundance isotope ratio, which is like 1%. But this is not actually a problem anymore because NMR's probes and spectrometers have evolved the last number of years, or last 10-15 years. So now you can actually do a natural abundance methyl NMR spectra in, well, minutes to hours, quite a lot of minutes though, but in any way. And this actually makes the whole gives NMR the possibility to actually be used for studies of higher order structure, because there are applications such as batch-to-batch consistency. If you have originator molecule like a antibody which is available commercially and you want to compare your biosimilar, you can run these types of experiments, and it can also be used to do formulation optimization. And as I said, and one of the advantages here is that NMR spectroscopy has atomic resolution, and you can basically, it's also very flexible in the terms of what molecules you can put into your sample. So if you have a biological molecule, which is in formulation, you can directly put that into the NMR tube and study it. This literally means that you can take the sample that you inject into a patient and also study it with NMR. Of course, if you can have not assigned spectra, it's not, if you have assignment and knowing exactly which atom is more problematic, but you can still make quite interesting deductions just by looking at where specific amino acid types are in the spectra and correlate these to structure. And there is actually, for this, when you select the right type of method, you can actually study intact antibodies, which has a size of up to a size of 150 kilodalton. So how do you analyze NMR spectra? Well, the standard way is visual inspection, but it requires some form of NMR background to see what is significant. Of course, you can go pick peaks and then look at chemical shifts and integrations and things like that, which is also requires some experience. Another method, which is something that we do more when it comes to higher order structure, is we take the 2D spectra and linearize it, and then we can either just plot the intensities as a correlation plot, but you can also extend this for multiple samples into chemometrics and do PCA and clustering methods. And the whole validation of the method for studying MAB, there was a MAB study was published which was coordinated by the NIST Institute in the US, where the NIST Fab was studied, where they basically used the same experimental protocol over a large range of spectrometers in the world. We at SARomics acquired data sets for a commercial customer here, and we collected them using the NMR spectrometers at 800 and 900 megahertz at the Swedish NMR Center, and we had very good support from the staff at the Swedish NMR Center. But the result of this study is that for higher order structure similarities, to compare that if you have your antibodies the same, for instance, comparing different antibodies across different strain production units or batches, it is a very reproducible method and overlaps as well. So we made a case study of this, which we did in collaboration with Genovis, which is a Lund-based company that mainly works with, whose main business area is selling enzymes that modify antibodies in different ways, glycosylation or cleavage. What we did in this study was that we took the infliximab originator and the biosimilar, cleaved it, and then we both looked at the Fab and the FC domains, as well as the Fab and the FCs alone. We also did an oxidation study where we exposed the antibody to, so we could look at how the higher order structure changed with oxidation. So here is an NMR spectra of infliximab. It's not, and in this case, it's the cleaved because we wanted to use the Genovis enzyme which cleaves very specifically. And if you don't see any difference between the two spectra which are visible here, it's because they are very similar. And this is what we would expect, because the biosimilar is designed to be as close enough to the originator as possible. It's quite complex things, but you can pick out interesting things there just by knowing what to look for. One of the things we note when we did is that there's a large variation in peak intensities, and that indicates that this protein has some form of dynamical process. And both the originator and the biosimilar has the same dynamical process. And this has been correlated with other biophysical methods, which shows that what we're seeing here is a clear evidence of dimerization. But NMR sees this in a single spectra. But the main takeaway from this spectra is that the peaks are almost perfectly overlaid, which means that we have the same higher order structure for both samples, and we have the same three-dimensional structure. You can, of course, then deconvolute. If you have an antibody, you can deconvolute it by cleaving it into its constituent domains, which are the Fab and the FC. And then, of course, you can start doing making some detailed studies into specific atoms, because you know where the methionines are, and you know where the isoleucines are in the spectra. And you also know where they are in the protein structure, because nowadays with AlphaFold, we know where things are located. And so we can actually start to going down and seeing if we see two methionines in the FC domain and we see two peaks, we know which peaks they are. So we're basically getting down to atomic resolution of a 150 kilodalton protein. So you can actually get a lot of detail without even having the assignment and not even having to label your protein. And of course, you can do formulation studies or degradation studies here. In this case, we did a forced oxidation where we exposed the Fab FC in this case to peroxide, and we can see that the methionines are oxidized as expected. But what we also see here is that the higher order structure of the protein the peaks are the same before and after oxidation, with the exception of methionines, which tells us that the overall 3D structure is the same after the oxidation. We see small peak shifts for the isoleucine, but the funny thing about that isoleucine is that one is located right next to the methionines, or there's an isoleucine right next to the methionines in the protein structure, and therefore we can basically say, oh, this should be this isoleucine. So this is one thing you can do, forced oxidation. You can then take this even further, and which we did for the FC domain. And here we used a little bit more advanced NMR techniques, which was where we can record these spectra with a much shorter time, basically like less than 1 hour for a spectra, depending on how much signal and how we want to do it. We had good help from the Swedish NMR Center in how to test out and apply this method. And what we did was here, basically we exposed the protein to peroxide, and then we took spectra at different time points. And we could basically see that we could follow the oxidation of the methionines, and we see that the two methionines are oxidized in different manners, where you still have one of them in intermediate thing after 2 hours. And then we can take this even one step further, would be to actually look at the oxidation products. Because when you do oxidation of methionines, you basically add 1 or 2 oxygen atoms, but the methyl group remains, and that's still something which is detectable at NMR, but at a different chemical shift. So what we could do in this type of experiment is that we could also look at how the oxidation products appear in the spectra, and we see that there's quite a different oxidation profile for these two methionines in the FC domain. And with that, I think I'm basically going to summarize it and say, like, NMR spectroscopy is a tool for characterization of biopharmaceuticals. It has atomic resolution in solid solution state. And I think one of the strengths is that it's quite flexible. As long as your biological molecule is soluble, you can basically study it by NMR. You can even do something which is a formulation. It is information rich, but it's sometimes hard to interpret the information. We use it for our higher order structure characterization, where you can do easy validation of the higher order structure by comparing originators of biosimilars. And this can be extended into chemometrics. And we can detect dynamic processes easily. And from other collaborations we have with academics, I can say that NMR is very much very good for adding detailed information in correlation with other biophysical techniques. And with that, I would like to end this talk.